All right, on behalf of the IU Health Bloomington Hospital Medical Education Department, we would like to thank you for attending today's medical lecture. Recording attendance is pertinent for us to be able to continue the CME program. Please take note of the directions listed on the screen for attendance, obtaining CME credit, and CME recordings. Today's presentation is about immunizations and the COVID vaccines by Dr. Tom Hrisomalos. Dr. Hrisomalos practices at IU Health, Southern Indiana Physicians, Infectious Disease, Bloomington, Indiana. At the end of the presentation, Dr. Hrisomalos will gladly take questions via the chat box or by unmuting your mic to ask a question. We'll go ahead and turn it over to Dr. Tom.
All right, can everyone hear me okay?
Yes.
Excellent. So, what a crazy time we've all been through here. And I thought with the next round of craziness that we're talking about here, trying to immunize the entire country for COVID, it's a good time to talk a little bit about immunizations in general. Then I'm going to talk a little bit specifically about COVID, and then go through the COVID vaccines, how they work, and some issues that have to do with these immunizations. So I'm glad to answer questions, and we'll try and do this in an efficient way. So why do we bother to immunize people? As an infectious disease physician, it is dramatic to me when you look through the history of immunizations and vaccinations that with the exception of safe water, not antibiotics or anything else has made such a dramatic improvement in mortality and population growth than immunizations. High mortality, high morbidity illnesses are still here. I always speak with people and they say, oh, I don't hear about anyone getting tetanus. I don't hear about anyone getting diphtheria or whooping cough. The tetanus bacteria is in the soil. It doesn't go away. And the diphtheria bacteria is in our population. We just don't get sick because we have antibodies against the toxin it makes. But the infection and the bacteria is still here. And if we stop immunizing, these things will reemerge very quickly. The economic costs associated with these illnesses are huge, let alone the indirect costs when you have a sick child missing work or when you are ill missing work are huge as well. I mean, just think of this in terms of the COVID issues now with, keeping kids out of schools and classrooms closed and all the costs associated with that. So immunization is a very cost-effective intervention. This graph here to the right shows cases of vaccine preventable diseases with and without immunization. This prior to immunizations being available, and this was an estimate around 2000, around 1998. And you can see without immunizations, there would be hundreds of thousands of cases of measles, of diphtheria, of pertussis, and other diseases that we just don't see. So the effects of immunizations are really, really dramatic. This slide shows the reduction in the number of cases and in the number of deaths before and after effective vaccines became available for these different diseases. So virtually 100% reductions in diphtheria and measles, 97% reductions in mumps and pertussis, 100% reductions in polio and rubella, let alone the deaths associated with these immunizations. Smallpox, of course, has been eradicated as a disease that's transmitted in the population. Tetanus with 99% reductions in fatal cases, 90% reductions in hepatitis A and hepatitis B and Hrisomalos and all these other diseases. Dramatic improvements in the number of cases that we see. So the immunizations are very, very effective. Now, what everyone has been hearing about is we have to achieve herd immunity when we vaccinate people. And let me say a few words about these things. We don't use all vaccines to create herd immunity. Some we do, and some we use for other reasons. So the idea between herd immunity is that if you vaccinate most of the herd, most of the population, the disease will disappear, and individuals in the population who for some reason can't be vaccinated will be protected because the disease will not transmit in the community. This would be an example. Polio vaccine would be an example. You immunize everybody and you get rid of the disease. So polio was eliminated as an infectious disease in the northern hemisphere probably 30 years ago, and we don't have any cases here as a consequence of that. You can also vaccinate people not to achieve herd immunity, but to vaccinate a susceptible cohort. And these are vaccines that are indicated in specific populations to protect them from disease. And an example might be giving meningococcal vaccine to college-age kids. While they're in college, they're at higher risk, and we want to protect them during that period of time when they're at risk, but we don't necessarily have to use some of these vaccines to vaccinate everybody. We're just picking out the susceptible individuals. Once a population is protected by herd immunity, then even small numbers of people who remain susceptible are protected. But if vaccine rates decline, herd immunity then is lost and people become infected once again. This graph over here shows cases of measles correlated with measles, mumps, and rubella vaccination in the United Kingdom. And as vaccinations rates declined there in the early 2000s, the cases of measles just went up. This vaccine level down here in 2000 to 2010 or so was simply below what was necessary to achieve herd immunity. And so the measles cases began rising again. And so that's the constant concern with many of these diseases. It shows a couple things. It shows that measles is not gone. It can be reintroduced into a population easily. And it shows if you don't maintain immunity, these things will come back. If you look at measles in the United States, we have achieved pretty good herd immunity. Here are the cases of measles, in the 1930s, 40s, and 50s, averaging 500 to 600,000 cases a year, dramatically going down with measles vaccines being licensed in the early 1960s. Then this smaller graph shows little bumps in measles cases all the way up here until 2019. And this is interesting because this is occurring still in the face of pretty good vaccine levels in the population, over 90%. But small little groups and populations are unvaccinated, communities that oppose vaccination and so forth. And we're getting outbreaks in these little bubbles of areas where vaccines are not being used to maintain that level of herd immunity. 2 doses of measles vaccine offer 100% protection. And if you don't get vaccinated against measles, your lifetime risk of getting measles is very, very high. So dramatic benefit of vaccines. People always want to know, well, how much, and we've discussed this a lot with the COVID vaccine, how much of the population do we need to vaccinate to protect ourselves against community spread? What's the herd level of immunity that we need to achieve? Well, it depends upon how transmissible an infection is. So measles, which has an R0 number of 12 to 18, that means for every measles case, 12 to 18 other individuals are infected, requires a very high level of herd immunity to stop it from spreading in a population. Diseases that have smaller R0s, such as mumps, you don't have to achieve quite as high a level of herd immunity to stop its transmission. Influenza is down here at 1.5 to 1.8. For every influenza case, 1.5 to 2 people get infected and acquire influenza. And so again, the herd immunity threshold becomes lower and lower depending on the threshold, depending on how transmissible the disease is. And it looks like the SARS-CoV-19 that we're dealing with right now is more infectious than influenza, but less infectious than, for example, the SARS of 2002 or mumps and some of these other infections that we deal with. If you have time, it's an interesting thing to visit this website. This is a history of immunizations, and it goes back through the history of when different vaccinations were tried and when different types of immunizations were tried and what people tried to do to prevent these diseases. I pulled off some citations out of this website. In early China and India, they would inoculate children with smallpox by grinding up smallpox scabs, blowing the matter into the nose or scratching it into the skin. Some people describe that this procedure may have started as long as 200 BC. The first English variolation where they did this smallpox procedure was in 1721, when Lady Mary Montagu brought the practice to England and had her 2-year-old daughter variolated. The issue with this was that sometimes it was fatal. 2 to 3% of those who had this procedure would die of the smallpox, whereas 20 to 30% would die if you actually got smallpox in the community. So the other concern about this is that people who were variolated could pass the disease on to others. And so there's an interesting history. A farmer named Benjamin Jesty in the 1700s inoculated his children with cowpox, and is attributed to be the first person known to introduce the cowpox inoculation to prevent smallpox. We've had issues, of course, in healthcare workers about mandatory vaccinations. You have to have an influenza vaccine to work and so forth. That's not a new thing either. 1777, George Washington ordered mandatory inoculation of troops with smallpox if they had not survived a smallpox infection earlier in life. And so the history is fascinating if you have a chance to go through those things. Vaccines in general have excellent safety profiles, but they are not devoid of adverse side effects, and they are not 100% effective. I put this one picture up here because this is a fascinating story as well. Pasteur developing rabies vaccine, and you can see a rabbit here on the table. And he infected rabbits with rabies, and then he killed the rabbits and extracted the spinal cord tissue, and he inoculated individuals with 2-week-old dried spinal cord, and then 10-day-old, and then 8-day-old, and then 5-day and 7-day and 5-day and 3-day-old spinal cord, going from what was probably inactivated dead virus, perhaps even to some live virus towards the more end of it. And that series of vaccinations would then protect an exposed individual who had been bitten by a rabid animal from getting rabies. So quite remarkable. So these are the common vaccines that are used for routine immunizations. Adults commonly will get these listed over here. Vaccines against shingles, hepatitis, whooping cough, mumps, tetanus, pneumococcal disease, influenza, measles, mumps and rubella, chickenpox, papillomavirus, and so forth. And so we focus a lot on these, and this is going to serve as a model as we go on to discuss COVID vaccinations. Bacterial vaccines come in all different flavors. You know, people have been concerned about these COVID vaccines and what's an mRNA vaccine and how is that different? Well, we use all kinds of different vaccines right now that work in different ways. So with respect to bacterial vaccines, for example, we have capsular polysaccharide vaccines where the carbohydrate capsule of the bacteria is used to generate an immune response. We have toxoid vaccines where we use the toxin that is produced by a bacteria to generate immunity. The toxoid vaccines do not prevent you from getting an infection. They prevent you from getting sick. They prevent the manifestations of the toxins produced by the organisms. But people who have had vaccination against diphtheria, for example, can have diphtheria organisms in their throat. They just will not become ill because we have antitoxin antibodies circulating around. There are killed bacterial vaccines, older cholera vaccines, plague, rickettsia, Q fever vaccines are killed whole bacterial vaccines. We have purified protein vaccines where we take specific proteins from an infectious organism and use those to generate immunity. That's the acellular pertussis vaccine. They genetically inactivate the pertussis toxin by adding 2 amino acids to it that inactivates it. It maintains its antigenic characteristics, mixing that with some other proteins from the pertussis bacteria and create the acellular pertussis vaccine. We have live attenuated bacterial vaccines, BCG against tuberculosis, oral typhoid vaccine, tularemia, a new cholera vaccine against cholera, Vaxchlora vaccine. These are live weakened bacteria that are used to immunize people. And then, of course, we have all the passive immune therapies. We can give antibodies directly against toxin, against botulism, against diphtheria, or bringing it up to modern times against COVID. This is not bacterial, but I put this as an example. Convalescent plasma against that is a passive passive immunity. We have a lot of different types of viral vaccines. We have the live ones. So measles, mumps, rubella are live, weakened strains of those viruses. Varicella vaccine that are given to children. We have the nasal influenza vaccine, rotavirus, yellow fever, adenoviruses types 4 and 7 vaccines, the old vaccinia smallpox vaccine. These were all live, and these were weakened in some fashion and would generate very good immune responses. We have killed viral vaccines, such as against polio, hepatitis A, rabies, Japanese encephalitis, and so forth. We have purified subunit viral vaccines where we take proteins from these viruses and use those to immunize people. The hepatitis B vaccine is hepatitis B surface antigen, the surface protein of the virus that we use as the vaccine, not the rest of the virus, just that surface protein. Influenza, we grow influenza in chicken eggs, fertilized chicken eggs, and then what do? We purify the hemagglutinin and the neuraminidase, and we put those into a vaccine. The virus is not there, but just those proteins. So those are subunit vaccines. And then, of course, with the COVID, what we're dealing with now are these mRNA vaccines and adenoviral vector vaccines that we're going to talk about just in a minute. In general, live viral vaccines probably induce better immunity than killed or inactivated vaccines. They tend to produce higher antibody titers. They generate a more brisk cell-mediated immune response, but they also potentially have greater side effects. They can't be given to immunocompromised people, pregnant women usually, and things like that. So just the point being, there are a lot of different vaccines out there. All these vaccines, the intent is to save lives. The CDC estimates that if you take Americans born between 1994 and 2013, that all these vaccines will prevent over 300 million illnesses and over 700,000 deaths over the course of these individuals' lifetimes. But not everyone in the United States receives all these vaccines. And as you can see here in 2017, these are little places in the country where we've had outbreaks of measles, mumps, whooping cough, and other diseases. And over here on the right, I'd like to show this slide. This is the reported cases of diphtheria in the Soviet Union. And in right around 1990, when the Soviet Union or the USSR collapsed, they stopped their childhood vaccination program for a period of time. And look what happened to the incidence of diphtheria. It just skyrocketed. And it just shows you that these diseases are here. And if you're not vaccinated, there's the potential certainly to get them. So let me say a word about a couple of vaccines. Tetanus, something we don't see very much anymore of. We don't see much currently, but that's because we're all vaccinated against it. We've all had childhood vaccinations and booster doses. Tetanus is a pretty dramatic illness. Here's a child with tetanus, all the muscles in spasm and contraction, something that fortunately we just don't have to see with effective immunization. Tetanus is because the bacterium Clostridium tetani produces a toxin tetanospasmin that gets into the central nervous system and inhibits the release of inhibitory neurotransmitters, and all the muscles go into tonic contraction. The vaccine is effective. I'm going to show you some slides of some side effects here. Serious allergy to Tdap or tetanus, diphtheria, pertussis vaccine is about 1 in a million. So it's not 0, but it's pretty low. How about pertussis? We see pertussis in adults because they don't get immunized through adulthood very often, and they come in with coughs and so forth, but we don't see too much pertussis in children because the pediatricians do a very good job of keeping people immunized. But it causes whooping cough. We don't see this like we did in the past because of the effectiveness of these immunizations. Here's a child with whooping cough. I mean, it's just hard to listen to, and that goes on and on and on because what a serious condition that can be. So of vaccines, some of them can have serious reactions. The diphtheria, tetanus, and acellular pertussis vaccine, about 1 in a million results in a serious side effect. MMR, about 1 in 20,000 can cause immune thrombocytopenic purpura, less than a million a serious allergic reaction. And so you can see these things do indeed happen, and I'd never discount them that they can't happen. It's just that the vaccine, the benefits of the vaccine so much outweigh having these diseases around. I think it's certainly worth the chance. How about influenza? 1918 influenza pandemic. Here's the Camp Funston in Kansas with all these beds set up to take care of influenza patients that were so sick at that time. That doesn't look too different, does it, than New York City, the Javits Center that was set up to take care of COVID patients as well during this pandemic that we're going through now. So there's a debate. Immunization, is it dangerous like this? Sudden infant death, neurologic diseases, autism, asthma, leukemia, brain damage, or is it like this, beneficial and a positive thing? And I've got some videos here that I was going to show, but for point of time, I want to just show one of them. And that's the one over the debate over autism. The scientific studies continue to show that vaccines do not cause autism. There have been dozens and dozens with hundreds of thousands of kids in these studies, and they do not. But that myth persists, and it persists in large measure because Dr. Andrew Wakefield down here produced a paper in 1998 where he claimed that chronic enterocolitis and regressive developmental disorders were associated with MMR vaccine. That paper was retracted. He was discredited. He lost his medical license because of it. There was a lot of unethical behavior there. But this type of thing contributes to the vaccine hesitancy that we see now. I'm going to play you a quick video interview, and you can sort of see some of this vaccine hesitancy that we have to deal with, and I know that the pediatricians have to deal with as well. Let's see.
To know what is being injected into their child.
Call it the Jenny effect.
Without a doubt, in my mind, I believe vaccinations triggered Evan's autism.
Almost a decade ago, actress Jenny McCarthy became the spokeswoman for the anti-vaccination movement, a movement that's only grown stronger, finding support among liberal, well-educated communities, despite science showing over and over there's no link between autism and vaccines. In California, for example, a study out this week shows low rates of vaccination in San Francisco and Marin County, both wealthy areas. In Southern California, affluent areas in Los Angeles have had immunization rates that rival South Sudan's.
When parents choose not to vaccinate, other parents around them may have similar ideas. So what you get are pockets, groups of people who think the same. And so you now have a cluster of children who are susceptible, all kind of living and playing together, going to similar schools, houses of worship, and the like. So if that bad germ gets into that group, all of a sudden, you'll have an epidemic.
So why don't parents believe the scientists? Some of them are convinced the government is working with the pharmaceutical industry and just want to sell vaccines.
I am more willing to take the chance of her getting one of these, rare viruses or diseases and give her these side effects of these vaccines, which a lot of the times is autism.
I do not see that there is ever an acceptable time to inject a known toxin, a known poison, a heavy metal into the body of a 6-pound and up child in order to help save their life through a vaccine. No poison is safe. No poison can be given to a child, and it's okay.
The supposed link between autism and vaccines was championed by British scientist Andrew Wakefield, but his paper was discredited and redacted from the British Medical Journal in 2011.
The study has not arrived. The findings that we make have been replicated in 5 countries around the world.
Sir, that's not true. You've been offered the chance to replicate your study, and you've never taken anybody up on that. You've had plenty of opportunity to replicate.
Just giving you a false. I am telling you that this work has been replicated in 5 countries around the world.
That was not true, but even so, many parents still believe Wakefield was right, still believe the government is lying to Americans, and still refuse to vaccinate their children no matter what. Elizabeth Cohen, CNN, reporting.
So this type of vaccine hesitancy just plays into the difficulty that we're going to have in vaccinating large percentages of the population against COVID as well as against other diseases. Now let me spend about 5 minutes here reviewing where we are with COVID, and then we're going to talk about the COVID vaccines. So I think as everyone is aware, the COVID-19 illness that we're talking about now is caused by a coronavirus. These viruses are present in human and animals. There are 7 coronaviruses that cause disease in people. 4 of them here cause common colds. The other 3 here cause SARS, the illness that was an epidemic in 2002, MERS, the Middle East Respiratory Syndrome from 2012, and the current one that we're dealing with now. People are commonly infected with these coronaviruses, and these have caused the epidemic illnesses that we're dealing with. These are transmitted by the respiratory route, and here's pictures of them, the viruses over there. These viruses are the SARS-CoV-2 virus that we're dealing with now appears to have originated in bats, where a lot of these coronaviruses do exist. The full-length genome sequence of what we're dealing with is most related to a bat SARS coronavirus of this strain, 96% identical. And so that's the origin, and that's what we're dealing with. We know the symptoms of COVID-19, mostly fever, dry cough, fatigue, loss of taste and smell, headache, flu-like symptoms, with onset 2 to 14 days after you've been exposed. And about 80% of people who get COVID-19 have really mild to moderate illness. About 13% have more severe disease, and about 5 to 6% get critical disease, ending up in the hospital and intensive care units and at risk for dying. Here's a chest x-ray of a patient with COVID-19. The mortality of COVID-19 is somewhat hard to judge because it's hard to know how many asymptomatic cases there are. If you take symptomatic cases, the mortality is in that 2 to 3% range. With SARS in 2002, it was 10%. With MERS, it was 35%, so certainly better than those. If you look in all cases, the mortality is 1% or less, 0.5% to 1%, because there's a lot of asymptomatic cases. The major risks for dying with the COVID-19 is age. Here's studies from Geneva, from England, from Spain, all showing high mortality in the elderly population, much lower mortality in younger individuals. And this is the CDC's analysis of several different studies that show that if you are 65 or older, you have a 5 times higher risk of being hospitalized and a 90 times higher risk of dying than if you're younger. So you can see the risk go up with regard to hospitalization, and the risks go up with respect to mortality by age. So age is the number one thing that we worry about. If you look at age in comparison to other risk factors that we talk about, like hypertension, heart disease, diabetes, obesity, here are the risks for those, the increased risks for those medical conditions, and here's the increased risk for increasing age. So age is the dominant factor, even more so than diabetes, hypertension, obesity, asthma, and all the rest of the things that we worry about. So where are we at with COVID-19? There have been, I made this slide a few days ago, and I looked it up on the web, there had been 348,000 deaths in the United States from COVID-19, which is approaching the total number of combat and non-combat deaths from World War II, which was over, what, a 4-year period of time. Still significantly less than the 1918 Spanish flu, and the population of the US was about half of what it is. It was half then as to what it is now. But still 348,000 deaths, and we're still in the midst of this epidemic. The 1968 Hong Kong flu pandemic killed 100,000 people in the United States. The risk of death per year from COVID right now is about 1 in 1,000. About 1 person in every 1,000 people in the United States has died of COVID. Compared to yearly influenza deaths, about 1 in every 6,500. Deaths from being hit by lightning, 1 in 6 million. Deaths from a tornado, 1 in 13 million, and so forth. And so this is a significant illness that we're dealing with. I put these slides up there to emphasize that it is not the common cold and it's not influenza. It is much more serious than that. Timeline, just to remind people, the first cases that were described in December in China. The first United States case was January 21. The United States declared a public health emergency in February, and by April, 95% of Americans were under various types of lockdown orders. The first case of COVID-19 in Monroe County was in mid-March. This is March 22nd. This is the newspaper from March 20th here in Bloomington. It was May 1st that this antiviral drug remdesivir was given emergency use authorization. Dexamethasone, a valuable treatment, in July. And then bringing it up to December 11th, the Pfizer-BioNTech COVID vaccine was granted emergency use authorization. And then a week later, the Moderna COVID vaccines. And of course, these are the ones that we're giving now. So the question with these vaccines is, how do we end this? Here's a picture of one of the New York City hospitals dealing with COVID patients. So COVID virus uses this spike protein on its surface, here shown in magnification. It's a trimer. It's got 3 of these protein subunits linked together. It uses this spike protein to bind to the angiotensin II receptor on the surface of cells. It binds, it undergoes a conformational change that brings the virus in proximity to the membrane, allows it to fuse with the membrane and enter the cell. So our efforts with vaccination have been to try and develop an antibody that binds to the spike protein and blocks it from binding to the ACE2 receptor, and therefore the virus cannot get into cells and can't cause an infection. It's important to note that antibodies can be protective, we call that neutralizing, or non-protective, non-neutralizing. So just because you have antibodies against a virus doesn't mean that that protects you. The antibodies have to be against the correct part of the virus to do what we want. We need it to that part of the spike protein that binds to the angiotensin II receptor. I always tell people to think of it this way. People have HIV antibodies in their blood. That's the test that we use to diagnose HIV, but they're still infected with the virus. Those antibodies are not protective. They don't do anything to stop the virus. People who have hepatitis C have hepatitis C antibodies in their blood. We use that test to diagnose that they have infection, but those hepatitis C antibodies are non-neutralizing. They don't stop the virus from causing infection. So it's not just generating an antibody, it's generating the right antibody. And that's where a lot of research went into to develop these COVID vaccines. So here's a picture of the spike protein to which we're trying to develop an immune response to prevent infection. And I put in, I noted here these little blue areas from looking at the top of the spike protein, or the little blue areas here looking at the side view of the spike protein. These little blue areas are what bind to the angiotensin converting enzyme, the ACE2 receptor. So the ACE2 binding site is these little areas. So antibodies against these other areas may or may not be protective. They may or may not sterically inhibit the binding of the receptor and the spike protein. And that's where a lot of research and study went into to develop the vaccines that we have. So what happens when a coronavirus causes an infection? The coronavirus binds to the receptor, it gets in, it releases its RNA, its genomic RNA, which goes to the ribosome. The ribosome translates into proteins, non-structural protein. Several of these non-structural proteins, 16 of them, are made inside your cells that create a replication transcription complex, which will then take the RNA and make a whole bunch of other proteins inside your cells, proteins that comprise the capsid around the viral RNA, the spike protein, a membrane protein, an envelope protein. All of these proteins then go to the endoplasmic reticulum and then to the Golgi, coat the RNA. The virus is formed inside a vesicle, and then it buds off the cell and leaves. And I put this up here because I've had many people call me and say, oh, gee, I'm hesitant to have messenger RNA inside my cells to produce a spike protein. That sounds like something I don't want to do. But when you have a COVID infection, or any other viral infection for that matter, you have dozens of mRNAs and dozens of proteins being produced inside all of your cells throughout your body. That's the nature of an infection. The mRNA vaccines basically mimic this process without putting anything inside your cells that goes into the nucleus or interferes with your DNA, and nothing that is able to replicate or create a viral infection. So just remember when people worry about getting these vaccines, that the nature of all viral the nature of all viral infections is this replication process inside your cells, many of them inside the nucleus of your cells, in the cytoplasm, producing dozens of proteins oftentimes. And that's what we're trying to prevent. That's what we're trying to block. So another question I've always had is, well, how did we get this vaccine so quickly? And did they take any shortcuts? Because the normal vaccine pathway oftentimes takes years to develop an effective vaccine. Currently, there are hundreds of SARS-CoV-2 vaccine candidates in development. More than 5, but 5 I've got here on this slide, have made it into large-scale trials. And 1 has been approved for general use. 2 have been given emergency use authorization in the United States, the Moderna and the Pfizer vaccine. 2 have been given emergency use authorization in European countries and Great Britain, their AstraZeneca vaccine and the Pfizer vaccine. The one that's indicated on this slide is a general approval for a vaccine that China has developed and they're using in their population. So how did we get to this so quickly? When normally it takes years to get a vaccine, how did we get this so quickly? And it's because a lot of the steps and stages that are normally done to develop a vaccine were done concurrently rather than sequentially. And so the typical initial process in creating a vaccine is you study the virus, you learn about what's important about the virus, how it causes infection, what receptors it binds to, what is effective immune response in terms of blocking that virus. And we were very fortunate in that this coronavirus is very similar to the SARS coronavirus that caused the epidemic in 2002. And a lot of that research was already done. They understood this virus in great detail before it stepped onto the stage to cause disease. So a lot of that research was already done. We didn't have to start from scratch, so to speak. Then there is the issue of the phase 1 clinical trials, phase 2 and phase 3 clinical trials. And these were oftentimes started concurrently. where they did studies to look at the dosing and the safety concurrent with starting to vaccinate larger groups of people. And you can sort of see in this little box here, it's sort of half shaded and half shaded, because the phase 2 and the phase 3 clinical trials sort of overlap. They didn't not do any piece or part. They just sped it up by overlapping them. And then the brown area of this slide here shows the manufacturing of the vaccine. And that was dramatically started up. They took a risk. They said, we think these vaccines will work, so we're going to start manufacturing them. And they started manufacturing them early while the studies were still in progress. Normally, as shown up here, the manufacturing of the vaccines follows all these studies once everything has been shown to be effective. But money was put in by the US government and others to manufacture the vaccines before they were found to be safe or effective, in the hopes that they would be found to be so. And since they were, in the case of the mRNA vaccines, all that timeline was again compressed. So we have a vaccine now, you know, just at a year since this virus started onto the stage, and that's pretty dramatic achievement. So what about the different coronavirus vaccines? There are lots of different ones that are being studied. If you look at the different types of vaccines that are out there, there are DNA vaccines, pieces of DNA that are put inside your cells. There are RNA vaccines, and these are covered by lipid nanoparticles. These are the Moderna and the Pfizer vaccines that were given. There are just the protein vaccines, just purify the protein spike, the spike protein, and give that as vaccine. That would be sort of analogous to the influenza vaccines that we're dealing with now with the hemagglutinin and the neuraminidase proteins. Purify those and give those as an immunization. There are non-replicating viral vectors. They take other viruses that have been well studied, such as adenoviruses, particularly adenovirus 5, and they put the gene for the spike protein inside the adenovirus. They disable the virus so it can't replicate, and then they give that to carry the DNA into your cell. Adenoviruses are DNA viruses. They carry DNA into the nucleus of the cell to replicate, to produce, adenoviruses normally replicate, but they produce a messenger RNA that codes for the spike protein that will then generate an immune response. There are studies with replicating viral vectors that actually the virus actually replicates. There are studies with inactivated viruses. So China has taken this the SARS-CoV-2 virus, grown it up in the lab, inactivated it, and is then injecting that as a vaccine. And people are trying to develop weakened strains of the virus as well. So all these are out there. The candidates that we're dealing with are the SARS-CoV-2 inactivated. There's 8 of those. There's live attenuated, 3 of those being studied. The RNA vaccines, there's probably 20 of those being studied. So there's a lot of these candidate vaccines out there. So where are we at? In the United States, we have the Moderna and the Pfizer BioNTech vaccines given emergency use authorization. In Europe, they have Pfizer vaccine, and countries England is using their Oxford AstraZeneca vaccine. And then there are other vaccines that are approaching applying for approval. The Johnson & Johnson vaccine, for example, is coming up probably here towards the end of January, which is a adenoviral vector vaccine. So how do these 3 types of coronavirus vaccines work? The ones that I've been talking about mostly. The protein-based ones are just purified spike proteins. You purify the protein and you inject them. None of those have been approved yet. The messenger RNA vaccines are the ones that we have here in the United States, where the RNA for the spike protein from the virus is enclosed in a nanoparticle injected into your muscle. The messenger RNA gets inside the cell. It produces the spike protein. It doesn't replicate. It can't produce an infection. It produces the spike protein, and then you generate an immune response against that spike protein. Or the viral vector vaccines, where the DNA of the viral vector, like an adenovirus, is changed to have the gene for the spike protein in it, and the DNA of the virus is changed so the virus cannot replicate. Usually they damage a polymerase or something in the virus so it can't replicate. And then this virus is injected, it infects cells, the DNA is converted to RNA, the RNA makes spike protein, and you generate an immune response. So a lot of different ways to get to the same endpoint. Here's a list of some of the common vaccines that are out there. And let me just go through these sort of quickly. So the Moderna vaccine is a messenger RNA vaccine. It's messenger RNA in a lipid nanoparticle injected into your muscle. The messenger RNA gets inside the cell. That gives instruction to the cell to make the spike protein, which is then expressed on the membrane of the cell and generates an immune response. The Pfizer-BioNTech vaccine is similar. The Oxford vaccine by AstraZeneca is a weakened chimpanzee adenovirus to which a DNA gene for the spike protein of coronavirus is added, and early genes to replicate the virus are disabled. And that is how they carry the information to the cells for it to make spike proteins. There are others here. The CanSino vaccine is an adenovirus 5 vector vaccine. This one up here, made in China, is an inactivated coronavirus. They take coronavirus and grow it and kill it and add a adjuvant to it and give that as an injection. The Russian vaccine down here, Sputnik 5, is interesting. They've picked two adenoviruses, an adenovirus type 5 and an adenovirus type 26, viruses that cause common colds in people, and they've added the gene to produce the spike protein to those two viruses, giving the first shot being one of them, and then the second shot the opposite virus. So these trials have been going on for the past several months, involving tens of thousands of participants. The Johnson & Johnson vaccine, which is a adenoviral 26 vector vaccine, has enrolled 60,000 participants. AstraZeneca, 50,000. Pfizer had 44,000. Moderna had over 30,000. And so these have all, people have all been studying these vaccines. The only one that is currently being touted as a one-dose vaccine is the one by Johnson & Johnson. And they say that they will have data by the end of, January to present about their vaccine. So these messenger RNA vaccines are new, but they're not unknown. People have been working on mRNA vaccines for a long time. They've been studied for flu, Zika, rabies, CMV, and the nice thing about them is that once they've developed these lipid nanoparticles, they can put messenger RNA of anything inside them to be carried inside your cells. So it's easily adaptable from one viral pathogen to another once you understand those viral pathogens and know where it's important to generate an immune response. So messenger RNA vaccines cannot give someone COVID. They're not live. They don't replicate. They don't produce a virus. They just give instruction to the cells into which they're injected in your arm to make this protein for a transient period of time. The messenger RNA is rapidly degraded. It doesn't get inside the nucleus. It doesn't alter the DNA. It just gives the instructions to make that protein, and which generates a brisk immune response. Messenger RNA vaccines have been studied for a long time, trying to figure out how to deliver the messenger RNA to cells. And this is just a table that shows many of the different mechanisms whereby messenger RNA can be packaged to get inside cells. The vaccines that we have now use a lipid nanoparticle structure. So here's the Pfizer-BioNTech vaccine. The messenger RNA codes for the spike protein. It's encapsulated in a lipid nanoparticle. It's injected. The messenger RNA gets inside the cell. It tells the ribosomes to make the spike protein. The spike protein is then expressed on the surface of the cell, generating an immune response. They've studied it in over 44,000 people. It was given early emergency use authorization on December 11th. And there's a lot of detail about how the spike protein, how the messenger RNA is altered to make these spike proteins very immunogenic. I would just say that if you remember that little diagram that I showed of the spike protein binding to a cell, the spike protein has to have a particular conformation to bind to the receptor. Once it's bound, its conformation is altered. We don't need an immune response against the altered conformation. We need it against the original conformation. And so most of these messenger RNAs are altered so the spike protein becomes more rigid and cannot revert its conformation. So it retains the immunogenicity against the structures that we want it to retain, that we need for it to retain. The Moderna vaccine, mRNA 1273, has been studied in over 30,000 people. It was given emergency use authorization December 18th. And similarly, its messenger RNA packaged in a lipid nanoparticle goes to the ribosome, makes the spike protein that's expressed on the surface of cells and generates the immune response. These vaccines do have more side effects than your typical flu shot that we're used to. Here's side effects from the Moderna vaccine, and these are different doses. But I would just say after vaccine number 2, with the medium dose, which is what is being used, you can look, most people will have some moderate symptoms, arthralgias in say as many as 50%, fatigue, fever in as many as 30 to 40%. And so they have more side effects than some of the typical vaccines that we're used to dealing with. If you compare Pfizer and Moderna, the two vaccines that we have available, they're very similar. Pfizer is approved for age 16 and older, Moderna for age 18 and older. Both of them have effectiveness about 95% in preventing symptomatic COVID. Effectiveness in preventing severe COVID really is not known. There were 3 cases of severe COVID in patients in the Pfizer study. 2 of them were in placebo recipients, and 1 was in someone who got the vaccine. So they're saying that it's 66% effective in preventing severe COVID, but that was only 3 patients. So it may be much better than that. We just don't know. There were 11 cases of severe COVID in the 30,000 patients getting the Moderna vaccine. And interestingly, all of those were in placebo recipients. None of the people who got the Moderna vaccine had severe COVID. They seem to be equally effective in ethnic and racial groups. We don't know whether they prevent asymptomatic infection. There are 2 shots, 21 and 21 days apart. There are allergic reactions that probably have to do with the presence of polyethylene glycol in the vaccine. Side effects with the Moderna vaccine seem to be greater after the 2nd dose than the Pfizer vaccine. Headache, 39% to 63%. Fatigue, 51 to 68. Chills, 23% to 48%. But they're still quite similar. They have not been tested in pregnant or lactating women. The Pfizer vaccine has to be kept stored at very cold temperature, and it must be used shortly after it's thawed. The Moderna vaccine can be kept at a refrigerator temperature for as long as 30 days and room temperature for up to 12 hours, which is probably an advantage. But we don't know how long they last for. We hope that they last for at least a few years. Let me say a few words about the adenoviral vector vaccines, because there's several of these, and this is what the AstraZeneca and the Oxford vaccine is made after. So adenoviral vector vaccines are made by taking adenovirus, a common cold virus. It's a DNA virus. They take a DNA that would code for the spike protein in COVID, SARS-CoV-2, enter it into the adenovirus DNA. They disable the genes that allow the virus to replicate, and then they use that to inoculate people. The problem with these adenovirus vaccines seems to be that some people have pre-existing immunity to them. So this is a common cold virus, and we all have been exposed to this. So when you study people in the United States, 40 to 45% of people have antibodies against adenovirus type 5. And will that impair the vaccine from working? In sub-Saharan Africa, it's 90% of people have antibodies against that adenovirus 5. So people have been somewhat reluctant to use adenovirus 5 as the vector to carry the SARS, the COVID spike protein. And we call that vector immunity, when the immunity against the viral vector might impair the ability of that vaccine from working. And so how do you get around that? Well, these vaccine manufacturers have picked two ways. One, they have picked uncommon adenoviruses that for which there's not a lot of immunity in the population. So adenovirus 26, for example, about 1% of people have antibodies against it. And so they have used that, thinking that there will not be pre-existing immunity to impair the vaccine's effectiveness. The AstraZeneca vaccine from Oxford, they decided to use simian adenoviruses, vaccines that infect apes and monkeys, and they picked a chimpanzee adenovirus. These people would not have immunity to that. So simian adenoviruses are closely related, but they're significantly different in that immunity against the human ones do not cause immunity against the chimpanzee ones. And so they have been working with chimpanzee adenoviruses and have put different genes into them to carry those into people to provide immunity against different pathogens that they've been studying: malaria, HIV, influenza, Ebola, SARS, hepatitis C, rabies, and so forth. And so one of the reasons that they were able to develop this so quickly is had been studying it for years in terms of generating immune responses to a variety of different pathogens by using these chimpanzee adenoviruses. So the Oxford vaccine works by DNA into the simian chimpanzee adenovirus, disabling it so it can't replicate, injecting that into people. It carries the DNA into the cell. The DNA is converted to messenger RNA. The messenger RNA makes the spike protein, and then you generate the immune response. So here's adenovirus, these non-enveloped DNA virus that have been studied for a long time as vectors for getting DNA into cells, both in treatment of cancers as well as in prevention of disease by creating vaccines. This vaccine also has more side effects than most other vaccines. So here's the side effects related to the Oxford vaccine versus the meningococcal vaccine: more arm pain, more tenderness, more fatigue, more headache, more malaise, and so forth, more chills. And these vaccines just generate a stronger, brisker immune response than some of the other vaccines that we're used to. Just a couple other comments. I know we're right at our limit of time. This is the Sputnik vaccine that Russia created. And what they did is they took two adenovirus vectors. The first vaccine is adenovirus type 26, into which they put DNA for the spike protein of the coronavirus. And the second vaccine is adenovirus type 5. And their thought would be, if you generate an immune response against the vector, the virus that's carrying the DNA message to the cell after the first vaccine, that's okay, because the second vaccine is going to be using a different adenovirus to which immunity generated during the first shot won't inactivate it, and you'll get you get a good initial immune response and a good booster shot. So that's how they went about it. But all these are using the same kind of idea and the same kind of technology. Cost-wise, the Moderna vaccine appears to be the most expensive one at about $37. The Oxford vaccine is cheapest at about $4, the cost to make these vaccines. There are differences in storage temperatures and so forth, as we have discussed. So that's about these vaccines. The second question is, will people accept them? And this is a study that was done on November 23rd, and it looked at different countries. And the United States, the subtotal of people who said yes, they would get the COVID-19 vaccine when it was available was about 2/3. Great Britain was greater at 75%. France was lower at about 50%. And so, this is something we have to combat. We have to convince people, once we are sure, and I think with the data that is presented, we are sure they are effective. and we know what the side effects of them are. We want to get as many people vaccinated as we possibly can to put an end to this epidemic. When people are asked why they don't want to get the vaccine, most people are concerned about side effects. They think that the vaccine may not be safe, or the next most common reason is they just don't think COVID is that dangerous to their health. They may be younger people or not have chronic medical illnesses. So these are the issues. I think we have a pretty good handle on what the side effects of these vaccines are. A lot of people have been concerned about what about autoimmune diseases, about what other conditions, and we will know over time, and I'm going to make some comments about this here as we end, about what potential longer term side effects of these vaccines are. But I would put this to consider, is that this is one protein from a coronavirus. And we do know that these coronaviruses, the one that causes common cold, as well as the ones that caused SARS in 2002 and what we're seeing today, that we have not seen autoimmune diseases and so forth related to the infection. And so we wouldn't expect to see untoward effects related to the vaccination, but time will tell. So what percentage of the population do we need to immunize to get herd immunity so this epidemic stops? Well, SARS-CoV-2 somewhere in this range of R0 number, which means somewhere in this range of herd immunity, somewhere from probably 50% to 80% is where we need to achieve to get the epidemic to stop. Herd immunity, as we've talked about, refers to immunizing people so the pathogen cannot spread in the community. These little gray balls are people who have been immunized. Here's a case, they spread it to the yellow as the first round, and they spread it to the green, and they spread it to the blue. In an unimmunized population, that spreads very readily. With partial immunization, transmission is decreased, and with a high level of immunization, transmission stops because it just can't spread among the people who have had immunization. So currently, we are marching along the process of giving the SARS-CoV vaccine to people who have a high occupational risk or a high risk of death. So older age or middle age, or people who are in healthcare environments because they have high occupational risk, and we'll move from those individuals down to the general population. So phase 1A of the current immunization program in the United States is long-term care facility residents, high risk, and healthcare personnel, high risk of contracting the disease. Phase 1B, which hopefully will start here in the near future, would include individuals over age 75, non-healthcare frontline essential workers like police and fire, and then from there down into people above age 65 who have chronic medical illnesses and so forth. This plan, like we're using in the United States, is not what is necessarily being used in other places in the country. The United Kingdom, for example, has chosen to go by risk of mortality first, meaning the elderly get it in preference to anyone else. Individuals over age 65 in decreasing age brackets, including nursing home residents and clinically extremely vulnerable individuals before anyone else, including before frontline workers and healthcare workers and so forth. Some other countries have taken the opposite approach. Indonesia has elected to vaccinate workers, middle-aged working individuals, not the elderly, thinking that they are the population that is spreading the virus. And if they immunize those people, they'll help to terminate their pandemic. In the United States, we have 54 million adults over age 65. And if we could vaccinate all of those adults over age 65 in the next few weeks, we would dramatically decrease hospitalizations and death by over 80%, well before we achieved what we would consider herd immunity. So should we be doing that? And the answer is, we don't know, but these are public health decisions. And I think after the fact, it'll be an interesting thing to study different countries and how they went about immunizing their population. So these are just my thoughts. to end up this discussion, what is the world going to look like in this post-vaccine world as we finish immunizing people in the next few months? I think there's going to be thoughts and concerns and worries about the side effects of these vaccines. I mean, think of it this way. If you're vaccinating 2/3 of the population, there's a lot of illness and a lot of diseases that are going to occur in 2/3 of the population within weeks of receiving that vaccine. People are going to have heart attacks, strokes, cancer, Guillain-Barré syndrome, neurologic illnesses. And I think it's almost human nature that people are going to say, well, perhaps my illness was related to the vaccine I got. So I think that's going to be an evolving concern over time. And I hope we're able to study that adequately so we can dissuade unwarranted concerns and recognize valid concerns as we go forward. Question 2 is, will this virus disappear or become endemic? Given that this has spread worldwide, it's hard to imagine that this will disappear. It will probably persist in certain areas, and we'll probably need to maintain a high level of immunity against this virus so it doesn't cause epidemics and disease in the future. That raises the question of booster doses, and we don't know how long do these vaccines last. Everyone is hoping that they last for a few years, so you won't need a yearly dose of a vaccine. And I think that's realistic. I would expect that we're probably talking at least a few years, 2-3 years immunity following these vaccines, but we don't know. People are going to be following that now over the next months and years. Another bigger question is, how do we prepare for the next COVID-19? You know, we have had the 2002 outbreak of SARS, which was contained very readily and rapidly. We've had H1N1 influenza outbreak, pandemic, and now we have this COVID-19 pandemic. And I don't think there's any reason to suspect we won't have other pathogens causing pandemic illness in the future. So what do we need to do to be better prepared for the next one? And to end that thought, and I know it's hard to think that this is really a reasonable conclusion, but it seems to me that in a sense, we've actually been pretty lucky. We have a virus with a relatively low mortality, probably under 1%, versus SARS, which had 10% mortality, or MERS, which had 35% mortality. I mean, if we had something that's more deadly than this, that would cause much greater havoc. The transmissibility of this virus is significant, but it's not like measles and whooping cough, which would spread many, many times faster than what we're experiencing with this. And we were lucky in the sense that we have already done a lot of research on on coronaviruses because of SARS back in 2002. So we already knew a lot about this pathogen. What if we are exposed to a novel pandemic pathogen for which that research has not been done and we have to start from scratch? So those are my thoughts as we go into this post-vaccine world. Well, I'm sorry, I'm over about 10 minutes, so I'm going to stop right there. If there's anyone who wants to put a question into the chat, I'd be glad to answer. Or if not, I would I'd be glad to speak with you on a more individual way. So I've got a bunch of questions here. Let's see. mRNA vaccine should be safe for immunocompromised. Yes, it's not live. It's not replicating virus, so it should be safe. And in the immunocompromised people who did get it, they did not notice any adverse side effects. If you receive the vaccine, develop symptoms 3 days later, and test positive for COVID, is this a false positive, or will you still be able to get the COVID after receiving the vaccine? The PCR test for COVID detects the RNA of the COVID, and it would not be expected that this mRNA injected into your muscle would cause a positive nasal swab for COVID. So if you test positive for COVID after you get the vaccine, you probably just happened to get COVID before you developed any immunity. I'm in medical genetics in Indy. We're helping ramp up testing. At present, the RNA testing for COVID uses 2 sets of primers. Most use 1. So it doesn't seem that yes, COVID vaccine should ever get a false positive on a molecular testing. I think that's a very good point. Thank you. I think that's gives, from someone who knows the details of the testing, that's correct. The 2 sets of primers gives you additional security in that regard. Without a vaccine, how did the Spanish flu in 1918 end with restoration of normal everyday life? That Spanish flu took about 2.5 years to pass through the US population. And eventually it's believed that there was just a high enough level of immunity. So many people became infected that it was no longer able to spread. That strain, H1N1, is still around. It has mutated and it's not the same strain as present in 1918, but it didn't disappear. It just altered itself and is still present. Any comments on the variant strain of COVID-19? So there's been one described in the UK that it appears to be more transmissible. There's been another one described in South Africa, which has mutations in the spike protein. And so people are concerned, will that perhaps make the messenger RNA vaccines or these adenoviral vaccines not effective? And I think that's an unknown thing. I would think it would be unlikely because antibodies against, you develop more than one antibody against that spike protein, and you'd have to have all of them not affecting the binding or the fusion. And I would think that the vaccine would still work, but it's a very good question. I don't know. According to the IU Health email to schedule the vaccine, people with a history of anaphylaxis, life-threatening reaction have required treatment should not get the vaccine at this time. This is in evolution because they just didn't understand what was in the vaccine that was triggering the reaction. They didn't know, was it, some chemical that was used in the process of developing the vaccine? Do people who have allergy to shellfish, nuts, or antibiotics shouldn't get it? Who shouldn't get it? And it looks like from the most recent stuff that I've seen that people who are allergic to polyethylene glycol are the ones that have the higher risk of the anaphylactic type reaction, and people who have other allergies are probably not at risk. But I think that's keep paying attention because we don't really know. Ongoing trials for kids, yes. The vaccines are being studied in kids right now. And so hopefully they may have some preliminary data about that by summertime. And I don't know particularly about populations of high-risk kids, but I know that they're studying this in kids right now. I have been told that the vaccine information is being recorded in CHIRP. So you should be able to access your vaccine record there. Where are the concerns regarding future fertility coming from? I'm not sure. I've read those same concerns that people are worried about that. I don't know where that has come from because there's nothing that I've seen that would suggest any implication for fertility. The difference in the cold requirement apparently is because the different vaccines, the Pfizer and the Moderna, use different lipids to make up their nanoparticle lipid membrane. One of them, and these structures are fairly complex, and the Pfizer vaccine lipid envelope is less stable at warmer temperatures than the Moderna one is. So they're not identical lipid bilayers. I think the Moderna one, I forget how many different lipids it has, and the Pfizer has different set of lipids, and so they're just different. But that is an important characteristic. And I think what you said here, right, it is possible that Pfizer is being overcautious. There's some evidence that their vaccine is more stable than the initial cold requirements are indicating. The current recommendation for pregnant and lactating women is that they are not contraindicated to get the vaccine, that they should discuss this with their healthcare providers, but it's not anticipated that these vaccines should cause harm. Yeah, that's a good question. What's the R naught for the more contagious variant? I've just seen things on the internet that said that there was, I saw one report, they reported an R naught of around 3, which 3.5, which might indicate that it could be 50% more, 75% more contagious, but I don't know that as a fact. Is thimerosal used in any of the vaccines? So thimerosal is ethyl mercury. And thimerosal used to be used as a preservative in vaccines. And because of people's concern over mercury, it was taken out of most vaccines. I think the only vaccine that currently contains any thimerosal is the multi-dose vials of some of the influenza vaccines. But all the single-use doses are preservative-free and don't contain any thimerosal. So I don't think that's an issue, and that's not an issue in these new vaccines that I'm aware of. When should people who have had COVID get vaccinated? We don't know of any particular worse or problems with getting vaccinated if you've had COVID. 2%, I believe, in the Pfizer study of people who got the Pfizer vaccine had COVID. Had COVID antibodies, and so they had COVID when they got the vaccine, and they didn't notice any particular problem. If you've had COVID, you probably are at very low risk of getting COVID again in at least 3 months and probably longer. So there's probably no urgency to run out and get the vaccine, but you probably still should be vaccinated because we don't know how long immunity lasts. So we're recommending vaccine for everybody, even if you've had COVID, and you could wait 2 or 3 months after your COVID to make sure you're completely recovered and feeling well before you go and get the COVID vaccine. Are there currently any studies on the spread of COVID in the asymptomatic individuals who have had the vaccine? They are studying that right now. That's a big question. So if you are vaccinated against COVID and you have a very good chance of not getting symptomatic disease, can you get asymptomatic disease and transmit the virus? And it's a question mark. I think the presumption is that at some point, yes, you probably could get COVID and probably carry it, because we know that people who have had COVID, a very small percentage of them, can get COVID again, and many of them just will test positive with the PCR test and may be asymptomatic. The assumption would be, however, that if you get COVID after you're immunized, you probably will generate a very brisk immune response, and you would not be infectious for 10 days or something like that. You probably would get rid of that fairly quickly. But it's a good question, and I don't think we know. What are your thoughts about the British lengthening the time frame between the 2 inoculations? In Britain, because they're in the midst of another lockdown and pretty dramatic spread, particularly with this new variant, they are trying to vaccinate as many people as possible at the cost of delaying second shots for people who have already had their first shots, because the first shot appears to give you partial immunity. So with most of these mRNA vaccines, it looks like you have about a 50% decreased risk of getting COVID at about 10 to 14 days after your first shot. And then after the second shot, your risk goes, your protection goes up to 95%. So they're trying to get as many people protected as fast as they can. I don't know, from my view, that's not how the vaccines were studied. That's not the data that we have to look at. And I would be really reluctant. I would think that you'd probably want to stick with what works rather than venturing out onto a limb like that. I would wonder if you've been vaccinated once and you delay it too long, do you have to start again? Are you going to need 2 shots to get very good protective immunity? I guess the answer to me is I don't know. They're trying to deal with a difficult situation, and maybe they do have data that I'm not aware of that suggests that this is okay. But I understand the intent. They're trying to get as many people vaccinated as fast as they can, and I hope it works. All right. Well, thanks for everyone for listening. I hope that at least gave everybody some information. And I think we'll get this posted and the recording out, and people can look at it. And again, as I said, if anyone has any specific questions, please give me a call or text me. Glad to talk about these vaccines.
Thank you, Dr. Tom. We appreciate your time today. We want to thank you all for attending today's presentation. For attendance, please email your name and email address to IU Health Bloomington Continuing Medical Education Coordinator Pamela Vaughn, pvaughn@iuhealth.org. You can also email her if you would like to receive a link of this recording or have questions regarding obtaining CMEs or CME transcripts. Thank you and have a great day.